Welcome to Persist Grand Rounds. This is an online series of lectures hosted by Persist, covering topics related to advanced EEG analysis for improving outcomes in both neurocritical care and epilepsy monitoring environments. I am Marie Terrell, the Director of Product Management with Persist, and I'm here to introduce our speaker and topic for today's Persist Grand Rounds. Next slide, please. So through our customers, we understand the importance of investing in the EEG community through education, where expertise and knowledge can grow towards the utilization and implementation of EEG and quantitative EEG. The goal of Persist Grand Rounds is to provide a platform for those with clinical expertise on topics of advanced EEG analysis as a way to connect with the broader community to share their expertise and knowledge so that we can all learn and grow together. Each online lecture features an experienced epileptologist or neurophysiologist, and in this case, neurosurgeon, presenting on a topic of their choice as it relates to EEG monitoring and analysis. And you can visit our website to keep up to date with the schedule. Next slide, please. So the opinions and viewpoints of each speaker are solely their own and do not represent Persist or any other commercial entity or commercial interest. Speakers are not required to include Persist, its software, products, or services. The content presented was not prepared or edited by Persist, and Persist is not providing any form of compensation. And the next slide. So for today, we have Dr. Kai J. Miller, Ph.D., M.D., Ph.D. He is a pediatric and adult neurosurgeon who specializes in brain tumors, epilepsy, and deep brain stimulation. He manages patients in both the operating room and in the outpatient setting. His research involves measuring electrical brain activity with patients undergoing therapy with implanted electrodes and to understand brain circuit dynamics and develop new therapies. Additional interests are pediatric neurosurgery, epilepsy, brain tumors, neurosurgical stereotaxy, and large-scale brain dynamics, motor circuits, electrical circuits of psychiatric diseases, and research in cybernetics and brain-computer interfaces. In addition to his active clinical and research programs, Dr. Miller teaches graduate students and mentors neurosurgical residents. So for today, Dr. Miller will be speaking on the connection between the computational activity of neural populations and signals measured from implanted neurosurgical patients. And the learning objectives are to become familiar with the basic types of electrical recordings made from inside the skull to identify seizure onset regions in neurosurgical patients, to describe the basic phenomena found in these recordings, and then to understand hypothesized roles for oscillations in the brain circuitry of movement and how these can be connected to new therapies. So we are very lucky to have Dr. Miller with us today. I know he is an extremely busy person, and we thank you for being here. So please, Dr. Miller.
Thank you, Marie. That's a very nice introduction. I'm grateful to get to talk to the audience here, and I'd like to tell you primarily about research that was done during my graduate training and related to measurements made from the surface of the brain called electrocorticography. And then later I'll branch into some newer things that we're doing with stereo EEG. I have sort of co-development and research things that I do with a few different companies. No financial disclosure. My team is lucky to be funded by the NIH. I was a Van Wagenen Fellow. If there's any neurosurgery trainees or people that want to go into neurosurgery, I'd recommend applying for that. It's a really great opportunity to go work with top groups around the world when you finish your residency training. I'm going to start, like I said, talking about clinical electrocorticography. So this is still done, although it's not done as commonly as stereo EEG. And what it involves are these grids and strips of electrodes where the clinical scale, still the most common scale that's used, is separated by a centimeter from center to center. Each of these electrodes that you see here on the brain surface is capturing about 500,000 neurons in the cortical lamina beneath. So it's important to remember that because we're only sparsely sampling the brain surface, these are about 5 square millimeters out of every square centimeter, And because we only sample a portion of the exposed cortical surface, that really we need to have simple paradigms, and the way that we interpret our data should be relatively simple, or it should be a phenomena that we would expect to be relatively conserved across a wider range of the cortical surface and within the interdigitated brain areas that aren't sampled by our electrocorticography electrodes. But we can do some really interesting things because we're working with human patients. And we do have really high temporal fidelity, as I'll show you. So we can capture population activity in a way that is difficult to do with something like microelectrode recording. And we do have, in many ways, a cleaner signal than we're able to get with EEG, although I know that there's some, there are amazing breakthroughs happening that allow us to identify sources better and better over time from the scalp. If we do the most basic thing with these electrocorticography electrodes, what we can do is we can first just look at the voltage trace that we get when we have patients staring at a blank screen. And we can take the Fourier transform of those. And what that does is it translates the language of when something happens in the voltage trace to how often. And that's plotted on a power spectral density, where we have power here on the y-axis as a function of frequency on the x-axis. Then we give patients a cue to move their hand. And then what you see is a different power spectrum. And this was measured from just one site that happens to be active during hand movement. And what we can do is we can take power in different frequency ranges. And I think this is something that many people will be familiar with in the audience. And we can plot that spatially onto the array of electrodes that we have. And what we see is that there are these high frequency increases in power that are spatially very focal. And then there are low frequency, and by low, I mean less than about 40 to 50 hertz, decreases in power that are spatially very broad. And these really correspond with narrowband oscillations, these low frequency phenomena that we see. And these are the kinds of things that people have seen in the raw EEG and from raw intracranial recordings going back more than a century. And what we hypothesized and then later showed, and I'll show you an example of this, is that these high frequency changes that we saw, and people will often call these high gamma, but they're not actually oscillations. So we call them sort of broadband changes or power law changes. But what they mean is that there's no preferred frequency that we're looking at. So there's no oscillation, and it looks a lot more like a random walk in time. And because it appeared to be very focal, and because of some other reasons that I'll show you later, we thought this could be an interesting tool, perhaps, to map brain activity. And so this is from a paper back in 2007 from work that was done a few years before that. And what you'll see here is that if we have a patient and then we have somebody interacting with the patient, it happened to be a much younger version of me at that time, we can have them interact with the patient and then plot brain activity. And here we were just plotting on the x-axis, we were plotting electrode by electrode. And on the y-axis, we were plotting power and that high frequency that was above 2 standard deviations from a baseline period. And then on the brain on the right is plotting that activity post hoc. And of course, there are applications that have since emerged that let you do this plotting brain activity in real time now. But this was the first iteration of that. And this is what we found, basically, that if you capture this broadband high frequency activity, you could map local cortical function in a very robust way, and it maps directly to hand sensory motor area in the classic sense. And I'll show you this once again. And here I'd like you to just see these bars and this activity on the brain rapidly form. We could also do brain-computer interfacing, okay? And so for that, what we do is we would capture these high frequency changes again, and we would map the high frequency changes associated with one domain, let's say imagined hand movement, to right-left movement of a cursor. And then we would take high frequency power from another brain area that would be in the tongue or face part of the somatotopic representation. And we would map that to movement of a cursor up and down on the screen. And then what you'll see here in this video is basically the area that we were translating for right-left movement in the hand area of the homunculus, and the area that we were translating for up and down movement from the tongue area of the homunculus. And patients were very rapidly able to control these two-dimensional cursors. And of course, now we see a lot about brain-computer interfaces that are from intracortical recordings. and even some with intravascular recordings. Um, and this technology has really expanded since. But for these types of recordings, what we've found is that basically very simple maps that are made just from this high frequency power allow patients to get control, right? And I think that there will be future applications related to stroke rehabilitation, although I've been saying this for a long time. And I guess actually some of these companies do have this now, the intravascular BCI companies and people taking advantage of the new deep brain stimulation systems for things like stroke rehabilitation. And all this was based on just some intuition that the field had about these high frequency power being able to be related to very local brain activity. And when we come up with heuristics for why that might be the case, we can start to think about the underlying electrophysiology of these populations of neurons. And for electrocorticography, it's different than from stereo EEG, and it's also different from EEG from the scalp in the sense that you're measuring directly from the neuronal population, but the population that you're measuring from always has sort of the same spatial orientation compared with these electrodes that you have. And so if you're measuring laminar current dipoles, which a scientist named Mitzdorf showed that that's what you're getting when you measure these types of things. He showed that back in the 1980s. We actually found that these electrocorticography potentials for the broadband portion correspond to something called the power law. And so that means, it means specifically that there are no oscillations that are important. And you actually see that here with this linear fit on a log power versus log frequency plot. And these peaks that you see here are just a line noise from the wall. There are also other phenomena that we can measure in the electrocorticogram. There are event-related potentials, and these can be either from visual, things like visual or auditory stimuli, but they can also be from stimulating other areas of the brain and measuring the response at a particular brain area. So that would be cortical evoked potentials or subcortical to cortical evoked potentials. We have oscillations, and these are the types of things that people have focused on primarily in the EEG and electrocorticography and now stereo EEG signals for a very long time. But the thing that seemed to be most specific for local cortical function is this random walk activity or this broadband spectral activity that is captured most easily at high frequencies where you don't generally have oscillations. Now, we know that there are oscillations at very, very high frequencies that we'll see in some pathological and non-pathologic states associated with Parkinson's or HFOs, for example, in epilepsy. And so those are typically not seen as often in electrocorticography. And actually, when we see these very, very high frequency oscillations, when we see these oscillations at high frequencies, they tend to be above the typical range that we're measuring our signal at, because our signal will run into the amplifier noise floor before you get to that high of a frequency. And for anyone that's interested in the manuscripts that the things I'm showing you came from, there are QR codes on a lot of these slides, and those will just go directly to, those are links directly to the manuscripts. So if we start out by thinking about this broadband spectral change, what I told you in the beginning was a hypothesis, but here's a way that our hypothesis naively fell out of the data. So what we did is, this is an example where I had patients performing repetitive movements of the hand. So movement of the thumb, then the index finger, then the middle finger, and then resting in between. The sequence of fingers was randomly chosen. But if you look at the changes that you see, what you see is that for certain areas, you have increases in this broadband power. And it's hard to look at this, and the white spots here are just the influence of the line noise, which is just the electrical signals produced by wires running in the walls and that kind of thing, that we've omitted at 60, 120, and 180 hertz. But what you see above about 50 hertz here is that there are increases in power across the entire frequency domain. And that really matches that hypothesis well, that what we're seeing is not an oscillation. But we can actually explore back down to low frequencies by leveraging the fact that we have many, many samples of these signals, and some features will vary a little bit independently from one another. And we can use that little bit of independence to strip them apart. And this is how we did that. So first, if you plot low frequency power versus high frequency power, and you plot samples of movements of the thumb here in red, of resting here in black, and then movements of other fingers in gray, you sort of see this general phenomena that the with movement of the thumb here compared with movement of the rest, so the red distribution versus the black distribution, you have an increase in power in this high frequencies and a decrease in power in this low frequencies. But if there's a phenomena that's going on that's a superposition of things that are happening across both ranges, then you wouldn't just plot one frequency for another versus another. You would actually plot them on a rotated axis. And so if you perform a principal component analysis, it sort of tells you the directions these change in. And what you're seeing here with these bar plots is the average of rest is the origin of the axes. And so, you know, for this low frequency feature, movement of the thumb or other fingers is a decrease compared with that. And then what we see in this rotated space is that there's a phenomena that changes for both the fingers and the thumb, and that along one axis compared with rest. And then there's another phenomena that changes specifically for the thumb along this different axis. And this is just performing a simple rotation in this feature space. But what's interesting about that is I didn't have to apply it just at, let's say, two frequency bands here, a low frequency and a high frequency range. I could actually do that at all frequencies. And when I do that, rather than getting sort of axes that are rotation in just two dimensions, you have axes that are rotations at every frequency band. And the first principal components of these, and this is after the power spectrum have been normalized, is something that's essentially flat across all frequencies. And that's here in pink. And if we plot that back to the frequency domain, what you see here on the left is an increase across all frequencies. And then on the right, you see that there's this low frequency decrease is captured by something completely separate. And so this shows us that a naive decomposition that knows nothing about what any phenomena should be, or even the decomposition itself doesn't know which is thumb and which is index finger and so forth during the process of stripping these things apart by their variance. But it naively reproduces that shape where we have an increase across all frequencies. And for those that are interested, there's a manuscript there. And if we try to make basic neural models for these populations of neurons underneath each electrocorticographic electrode, we can have very simple sort of neural, what we call it like a point model or something like that for what a single neuron would be expected to do, but what should generalize across the population and produce our signals. And there are a lot of different models that would be consistent with this, but I think all of them will have something in common in that they'll be modeling increases in firing rate. And when increases in firing rate are modeled, which is a Poisson process, it has some random nature to it. There's no special time scale. That's what produces these broadband increases. And this is, I think, the simplest model. And that is that when you have action potentials arriving at one of our neurons underneath our electrodes, it induces current influx, and that has a particular shape and time. And pulling charges in and out of these neurons are what produce, and having the charges then flow in and out at different layers of cortex is what produces these large scale potentials that we measure with electrocorticography. So if we have these charge influx and then they flow passively towards the cell body, or even if they flow actively, as we know happens with calcium gated channels, then you have a dipole that's set up here. And the fields that you would model with that dipole, the field potentials that you would model, basically reproduce exactly what we see with our fits that we did. And this is on a log-log plot, and this would be the linear range that you saw earlier that corresponds to that power law here at high frequencies above about 70 hertz, which is what we see experimentally. Now, of course, we built a model and tuned it to fit the parameters that we actually see in model synapses, and that reproduces our results. But there are a lot of potential models like this that would produce the types of things we see. I think all of them would have the implication that what you're getting when you look at these broadband spectral changes is something that's like a real-time neural population spike raster, or a real-time measure of neuronal activity. So that if you're looking at an electrode here from, let's say, primary hand motor cortex, and again, a thumb movement electrode, this is the central sulcus in yellow, Sylvian Fisher in green. It's precentral gyrus, that little knob there is the hand knob. You can look at the positions of the fingers measured here from a data glove, and you can compare those from times when you had movement to rest. And you see that phenomena that I just showed you. And you can also isolate different frequency ranges and see what happens with them. Or you can even isolate extracts of different frequency ranges, like that broadband process. So if you look at the oscillation change here, you see that it's spatially distributed throughout the pre and post central gyrus area. or the Rolandic area, and that there's a decrease in power associated with that. And then if you look at, and in here we're looking at this electrode that you see there, which is here. And then if you look at this broadband spectral change, you see the timing of this is very, very precise for just the movement of the thumb and not for movements of the other fingers. And that maps of this broadband spectral change for thumb movement versus rest are very sparse, right? There's just one precentral gyrus electrode and one or two postcentral gyrus electrodes. And we can do that. I did that for the thumb in that example. This is a different patient. And here you see the broadband change. It's very focal. The beta oscillations that we find often in perirolandic areas, there are decreases that are very broad. If we have patients move their index finger, we get activity, and these are not the same two electrodes. These are separated by 1 centimeter from one another. And so you see that there's very sparse and somatotopically specific representation of the individual fingers when you capture this broadband spectral change. But when you get these low frequency oscillations, they're very highly overlapping. So if we develop overlap metrics to quantify how overlapping these things are, we can compare, let's say, thumb representation versus index. We see that the beta oscillations are almost completely overlapping, while broadband is not really overlapping much at all. And the same is true if you compare the other pairs of fingers there. And we can see this pan out in these time courses that we generate. Here there's a site. The blue here is the thumb trace from the data glove. Pink is this one site here for thumb. And then we have another for index and another for little finger. So you see that I used to say little finger all the time. That was before Game of Thrones. But there's this really nice tight representation between the activity of the brain and the behavioral parameters that we measure from our data glove. We also find other evidence for local cortical activity being represented by these broadband spectral changes or broadband spectral changes captured at high frequency. And this is the work of Dora Hermes and her PI, Nick Ramsey, from when she did her PhD. I was lucky to be able to work with them when they did some of this stuff. And what they did is they had blocks of rest and movement. Here they're moving single fingers. You can see the representation that they generated in these patients from doing fMRI before they their surgeons implanted electrocorticography electrodes. And then they could compare the electrophysiologic changes they measured from electrocorticography electrodes directly on the surface of the brain with the changes that they saw in fMRI in those exact same spots in the brain preoperatively. And you see a very tight correlation between the bold signal of fMRI and that broadband spectral change. And when we compare the distributions, you see that really the broadband, but not the oscillations, are very tightly coupled to the regions of peak bold signal, whereas there are some outlying regions where you have bold signal changes in the fMRI that are weak, that seem to correlate better with the oscillations. So I told you before, what about the gamma rhythm or gamma oscillations? Why do people see it in data that you show them that appears to be narrowband? Well, the reason is this. You start out with changes that are spectrally very broad over all the range of frequencies, and there's an increase in power in those when you engage in some task. But at high frequencies, you lose your ability to resolve that because the amplifier noise floor sets the minimum amount of power that you can look at, because these things fall off so fast in power as you increase in frequency, they run into this amplifier noise floor and you can't distinguish them in the high range. And then at the low range, you have these coincident oscillations that are here. And so when you plot these versus one another, it looks like maybe you have a high frequency range and a low frequency range. But the truth is that you have something that's very broad across all frequencies. It's just that our ability to measure that robustly is lost because of both coincident physiologic phenomena in the oscillations and also our instrument limitations that we often run into with our amplifiers. And so when you look at these on spectrograms, you'll see something like this. And this is not a high frequency phenomena. It's really a broadband phenomena that's just most apparent in a certain high frequency range. Okay, so I hope I convinced you that broadband changes tell us about very local activity in motor cortex, and other papers will show that this is also true in regions that are involved with memory and speech and vision and auditory processing and so forth. But keeping us focused in motor regions, we can ask, well, what are the significance of these rhythms, right? So we have these rhythms that are spatially very broad, and they're not somatotopically specific, can we come up with some intuition for what it is that they're doing? So with the important caveat that before I begin, if I say beta oscillations here, I'm not talking about a general phenomena of oscillations that happen between 12 and 20 hertz and are true of changes that you might see in that frequency range in occipital areas or in temporal areas or in far frontal areas. What I'm talking about is specifically that 12 to 20 hertz oscillation or set of oscillations, because there do appear to be multiple that are overlapping, that are in primary motor areas. And I think anytime we talk about an oscillation in EEG or in electrocorticography or in stereo EEG or in local field potentials, we should keep in mind that these are not universal phenomena, right? These things don't exist independent of the brain's anatomy. And two things that share a time scale, there's no default reason why they should be related to one another. They may in some cases, but I think that there have been some very unfortunate blunders that have lasted decades in our field related to two regions interacting, because people say, well, they have a similar time scales. So then they develop very sophisticated hypotheses, when at the end of the day, there are simple instrument and reference explanations for the phenomena that are measured. And I think we need to be careful when we talk about these oscillations and rhythms, and we use these Greek letter shortcuts, that we're not talking about them as if there's some disembodied phenomena in the brain, right? They're only oscillations produced by certain types of tissue, and those tissues are idiosyncratic to specific brain areas in most cases. And they may be related to thalamocortical interactions in general, or they may not. And if that's the case, then maybe there's some link between them. But by default, we should assume that oscillations that we measure from different areas in the brain are not related to one another. Okay, so returning to these getting into these oscillations in more detail, we do want to know when we see an oscillation, and let's say we see it in adjacent brain areas, are those related to one another if we see them in adjacent electrodes? And so a metric we can use for this is something called phase coherence. And there are different types of coherences that people measure. I most often use something called the phase locking value that was developed by a French guy named Jean-Philippe Lachaud very long ago. And what you do is you take the voltage and you filter it for a specific range, okay? And then you normalize you can think about it as normalizing the amplitude, but what you're doing is you're extracting a phase time series. And I'm plotting from this site, that's my seed site here, 2 phase time series. And I can compare those with, let's say, a phase time series from an electrode that's 1 centimeter over. And if the phase time series that I get there is reliable at different points in time, then I can say that this region is highly coherent. Sorry, this site here is highly coherent with this other site. Okay, and I can even plot that as a function of the relative lead or lag between the two here on the x-axis, and then the magnitude of the coherence. And I can plot that both by the intensity of the color as well as the diameter of the electrode that we're looking at. And I can just plot that there. And this is sort of a vomit green large electrode that you have there. And then I can go to some other region over here. And here you see that the phase relationship is not stable over time. Okay. And so the coherence is low and there's a relatively large phase lag, but basically there's no real coherence here. Okay. And so I can then plot that spatially across the brain surface. So I have some seed site and I plot this now in these different regions. And something you may notice is it looks like there's very significant phase coherence across the whole array with this site that happens to be right there. But it turns out that if you do something called the common average reference, which is what I did here, then you're actually, and you have an oscillation that's present across a large range of your array, then there are a bunch of channels where you might not have that. And by taking a common average reference, you're inducing false coherence that is pi or 180 degrees, whichever you prefer, out of phase in all these other sites. Okay, and so we can actually plot that here. And if you sort these primary motor cortex index finger site coherences with these other regions, what you see is that in dorsal, meaning towards the top of the head, pre and post central gyrus, you have very high coherence. spatially with this index finger site, and you don't have that with the other sites, and they're all weakly negatively coherent, and that is a product of how I did my referencing. Okay, so that's an interesting phenomena. It says that basically we have these domains where we've identified function, and we've identified that you have broadband spectral increases, and you also have spatially very overlapping between fingers, regions of power decrease, and that these are phase coherent with one another. And there are a lot of examples of this. I don't show them all in this talk, but you can find them at this QR code link. We can also ask, well, these oscillations are there. Is there evidence that they're influencing local cortical activity? Okay, so we have an index finger site here, and we measure the voltages from that, and then we measure the broadband spectral change. And we can actually say, well, why don't we identify the phase of a low frequency oscillation here? In this case, I've bandpass filtered for 3 Hertz, kind of like how I showed you before. And I'm just denoting the instantaneous phase of this oscillation that I've measured here. This is using a wavelet in this case, by color, right? So every time it's at the peak of this trace, it's going to be pink, transitioning into yellow. And every time it's at the trough, it's going to be yellow, transitioning into blue. And then I can take my broadband change, and I'm actually going to use the log of the broadband because it happens to be log normal distributed, okay? And I can plot the log of the broadband here as a function of the phase of the low frequency oscillation. And then I can overlay these. So I take the individual sort of 1 cycle. You could think of this as taking the individual 1 cycle snippets of this broadband this broadband time course, and by 1 cycle I mean 1 cycle of the 3 Hertz oscillation, and I can overlay those with one another like this. And then I can take that overlay and I can calculate the average of the broadband power as a function of the phase of this 3 Hertz oscillation. And I see that there's a very strong sinusoidal relationship, meaning that this broadband signal change is very tightly coupled to the phase of this low frequency oscillation. And specifically, it's tied to about a -3/4 phase. And that's when the 3 Hertz oscillation is going from its trough and starting to rise again. That's when you're most likely to see broadband spectral change. And I'm sorry for introducing 2 complicated color scales in one plot, but I think it can be useful here. You can imagine rotating this, like this sinusoidal coupling plot here, and just plotting it with a color scale that now ranges just from red to blue with gray in the middle, and you get just one row here of a matrix that we call the phase coupling palette. And you can do that with wavelets that are from 1 Hertz all the way, let's say, up to 50 Hertz. And what you see immediately by looking at this is that there are multiple oscillations, and oscillations is a little bit dirty of a word here because they're smeared out over the frequency domain. It's not a very tight peak, but it's still somewhat peaked. There are very tight relationships between the phase of those things and this broadband spectral change, okay? And when we plot these palettes across the brain surface here, and here we're looking at the Sylvian fissure is along the bottom in yellow, and the central sulcus is along where my mouse is here. And you see that there's very, very tight coupling specifically to this 12 to 20 Hertz range that comes right in the dorsal Rolandic area, the dorsal pre and post-central gyrus area. This is not like coherence where there's some seed region. This knows nothing about that. It's just that there happens to be this oscillation that's there that's influencing local cortical activity. And then if you go elsewhere, there's a low frequency oscillation here. Again, this is actually down near 3 Hertz. That's very robust and topologically more specific to this. This is probably a inferior frontal gyrus region, getting down near Broca's. And then there's another oscillation down in the 6 to 7 Hertz range over here. And these things do have topological representation that can be relatively tight, okay? And we can actually capture these and see how they change with movement. So if I have one of these palettes, like I showed you before, instead of using wavelets at one frequency, I can collapse them across a range of frequencies. The technique that I use for that is something called the Hilbert transform. And it's described in this manuscript. It's well established. And I then capture coupling to this range of frequencies. And the phase of a range of frequencies is a little bit of a dirty term, but it's just basically imagining that you've bandpass filtered your signal. What are the times at which it's at its peak? Well, that's phase pi. And what are the times when it's at its trough? That's gonna be phase 0. So we plot our broadband power here as a function of the phase of this low frequency oscillation, as you see here. And we can create something that we call a coupling vector there. And it seems like I'm getting somewhat exotic, but there's actually a real use for this that's kind of fun. And that is that we can take our data now, and we can capture broadband, and we can capture these oscillations, and we can chop it up by what the behavior is, right? So this is times of movement of the thumb in green. This is the times of rest in dark gray. And then we can plot, for example, broadband as we had before. It's very high during movement of the thumb from this particular electrode and low during rest. We can also plot the 12 to 20 hertz oscillation amplitude. You see it's high during rest and it's suppressed during movement of all the fingers, and especially so for movement of the thumb. But we can also look at the interaction between these two phenomena. So we have the broadband spectral change coupling to the low frequency oscillation and revealed by this phase coupling vector. And what you see here is that the magnitude of this vector is very high during rest and very small during movement of the fingers. And we can plot all of these individual ones on a plot here in the complex plane. So this is 0, pi over 2, pi, 3 pi over 2. And that's reflecting what phase of this low frequency oscillation the broadband is strongest in. And you might be tempted to just take the amplitude of this coupling and then compare that from different states. But of course, something that's circularly distributed around 0 here should average to 0, not average to the amplitude of what it is. So we can project these things down onto the average vector for that task or for that behavior. So in this case, if it's rest, we plot it down here, and we can actually get a distribution here for phase amplitude coupling or rhythmic entrainment, or whatever you'd like to call it. I call it rhythmic entrainment during rest here in gray. And we can actually show that that is dynamically decreased during movements of any of the fingers, right? So we have these oscillations. We can show that they decrease in power and they are phase coherent, okay? We have broadband spectral change that's topologically very focal, but seems to be a marker of local cortical activity. And these oscillations are actually influencing local cortical activity. And they're doing it in such a way that they are suppressed during periods of movement. The oscillations and the influence of the oscillations are suppressed during movement. A question that I often get at this point is, well, is it just that the amplitude of the oscillations is decreasing during these periods and your ability to estimate the phase of those is diminished during periods of movement? And there's an element of that, but the decrease in entrainment is additionally significant beyond that. And the supplement of the manuscript goes into it. We can look at the overlap between these different phenomena and see that the basically it's the peaks of the fMRIs I showed you before really correspond best with the broadband signal change, but that the other areas correspond to these regions of where you have both beta amplitude and coherence, as well as the modulation or rhythmic entrainment. The interpretation for why I think we have this is basically that these oscillations, by synchronizing populations of neurons, right? So if we were to measure single units and the local field potential associated with these oscillations, what we're really measuring when we measure the electrocorticography and broadband are reflections of these, is that what we're seeing is the dynamic suppression of single neurons by forcing them all to be synchronized with one another, okay? And we called this the suppression through synchronization hypothesis. So that imagine, and this is, again, a little sort of toy heuristic here, but you have a synchronizing neuron, let's say a thalamic neuron that could be coming from, well, it'd be ventrolateral nucleus. For those of you that are functional neurosurgeons, you'd say it's, VOP or VIM generating this. And we know that these thalamic projections to motor cortex are projecting to GABAergic interneurons in layer 5, and those can have very coherent activity across the cortical surface. And so what they do is that they basically inhibit all of the pyramidal neurons in phase. And then when we have broadband increases, what we're seeing are complex representations because these populations of neurons are computing the things that they are created to compute. But if you synchronize them, you can basically make it so that the local activity of these pyramidal neurons, you can't have any complex representation because they are all forced to fire or be inhibited at the same time. When what you really want is a complex spatiotemporal pattern where these neurons are exciting each other. And that's what's reflected by the broadband. And so a diminished broadband and an entrained broadband, we think, is reflecting this suppression of cortical activity by synchronizing input. We think that this is related to the kinds of problems that you have in Parkinson's disease, right? Where we know that the amplitude of beta oscillations doesn't go down. And I'm going to show you some experiments that were performed by some colleagues that I was able to collaborate with that show us that there's something about this circuit that seems to be related to the rhythmic entrainment in Parkinson's disease, and our ability to interrupt this circuit in Parkinson's disease is correlated with the therapy. So here's some evidence for this. It's this idea that you have some kind of suppressive role for oscillations and that they actively entrain spiking activity that we measure by broadband, and that this mechanism breaks down in Parkinson's. And so there's a surgeon named Phil Starr, and he's at UC San Francisco. He's been there for a long time, and he did these really nice, he's pioneered these recordings that now a lot of people across the country do, where during DBS surgery, you slide a strip of electrodes back over the brain surface so they span across the primary motor cortex at the same time as you have a deep brain stimulation electrode going down to target. And that's what you're seeing here. And so one simple thing you can do is you can ask, if I look on the cortical surface here and I ask, well, does the timing of neurons in this circuit relate to local field potentials? Well, the simplest thing I can do is I can take a neuron from subthalamic nucleus, which is part of this cortex to basal gangliolothalamic circuit. And I can say, well, does the primary motor cortex activity relate to the timing of these individual units? And that doesn't mean that STN single units are causing primary motor cortex. It could be the other way, but you're looking for a correlation here before you try to establish causation. And what you see is that these oscillations immediately fall out. This is not a filtered signal. This is the raw signal averaged on the timing of single spikes here. And then this is the spectrogram averaged on the timing of single spikes. Okay, so here you see these broadband spectral changes. If you look across time, locked to the timing of a action potential in subthalamic nucleus, they are rippling. And the time scale that they're rippling on is this beta oscillation that we measure, or this motor oscillation that we measure. And the timing of single neurons from subthalamic nucleus establish that. It's a really cool effect. And this is showing the rhythmic entrainment in a little bit different way than I did. This is not accounting for phase, but it's instead looking at it's looking at power here with color and power at each frequency. And that's what you have on the y-axis as a function of phase on the x-axis here. And so you see that in Parkinson's disease, there are these high frequency changes that are capturing this broadband that are coupled to the phase of low frequency oscillations here in about the 12 to 20 hertz range. Okay, and it's much stronger in Parkinson's disease than you have for dystonia or you have for epilepsy. And these epilepsy patients were patients from Dr. Ogerman, and that was a collaboration between Dr. Starr and I was when I was Dr. Ogerman's student back then. And so you see that there's an enhancement of this rhythmic entrainment or phase amplitude coupling specific to Parkinson's disease. And then when you turn on the deep brain stimulation system, you see that basically this entrainment goes down. So that's causal proof that you can interrupt the cortical phase amplitude coupling that you see that is related to the timing of single units down in these regions by delivering DBS. So this is the likely mechanism by which DBS works in Parkinson's disease. And so you might imagine a new generation of deep brain stimulation systems. And I say this now, and this slide is back from from a time before Phil Starr showed that you can actually do this. So he went and proved that you can do this. You can look at phase amplitude coupling from these surface strips, and you can use that to drive the timing of stimulation and interrupt phase amplitude coupling with onboard closed loop systems like the one that's recently come out from Medtronic. So I think that's a really neat example of how you can identify these underlying physiologic phenomena, try to characterize what they mean, what they do, look at them in different behaviors, see how they interact with one another, and then ultimately see how they relate to specific disease states that we can treat. In this case, we didn't come up with a new treatment strategy. There was one there. It just explained why it worked. And then lastly, I'd like to move to more current things that we're doing as we've transitioned from electrocorticography to stereo EEG, which is really where the field has gone. And by the field, in this case, I don't mean the scientific field, I mean the clinical field, because the stereo EEG is much better tolerated by patients, and it really allows us to sample deep structures. Okay, we can also do things like radiofrequency ablation. We can do test stimulation with deep brain stimulation. That's what that page I got at the beginning of the talk was about a patient we would do this with. And when we're measuring here clinically, we're much more sparse now than we were with electrocorticography, but we're measuring from the volume rather than a surface. And if we want to ask, and the signals, by the way, are much different, as you might expect, in white matter and in thalamus than they are in cortex. And if we ask ourselves, what are the research implications of this? Well, first of all, the types of things where we look at hand somatotopy and all that, we can't really do that with stereo EEG that well because we just don't have the coverage. I only implant stereo EEG electrodes in the places where the neurologists would like to sample from to try to understand where seizures are starting and spreading to. And so it's not like we can just put tons of electrodes in motor cortex and look at functional representation or in early visual areas and so forth. It really depends upon any given patient's clinical situation where these go. But we can start to ask questions about deep structures that we couldn't before, right? Association areas at cortical areas along the midline, or basal ganglial areas if we happen to go through there, or thalamic areas in the cases where we happen to be sampling thalamus because we're considering doing chronic stimulation with deep brain stimulation to treat these patients' epilepsy. And we can study circuitry because we can capture multiple sites in a hierarchy because we can get to them. We can get to supplementary motor areas. We can get to deeper premotor areas in the parietal lobe and in the superior frontal gyrus and middle frontal gyrus that basically we're out of reach with electrocorticography. But we do have to keep this tissue heterogeneity and the signal heterogeneity that it creates in mind as we do this. So we can start. This is work from our team here at Mayo Clinic and a recent manuscript from one of my graduate students, Michael Jensen. Basically, these phenomena that we found in electrocorticography are recapitulated here in stereo EEG, right? So we give patients a cue to move their hand. They move their hand. And now, rather than timing anything based on cue, we just use EMG that we've measured. You can see the spectrogram here lines up very tightly with broadband spectral change being coincident with the EMG, as we might expect. And we can plot those over one another. The same phenomena really seem to emerge in the stereo EEG signal, at least when we're measuring from cortex, as we find with the electrocorticography. And here, we're mostly just capturing these broadband spectral changes now by capturing high frequency changes within our within the window that you have between the 60 and 120 hertz line noise produced by the ambient electrocontamination. And we can plot these changes on the slices of the brain rather than on the brain surface, as I showed you before. So we can look here at things like movement of the foot, the hand, and the tongue. And you can see that there's primary motor representation for each of these. And the project that I started Michael out on was sort of looking at these things. And we find some really interesting things. We have premotor areas as well that we're able to capture that are down here in the depths of the sulcus. And we have supplemental areas, okay? And we can do this not just in one patient, but in many patients. We can do the same functional mapping. And we can do it with speech, as you see here. And we can do this either by having patients just read the name of some visual stimulus that they see. And we can compare the audio trace. This is the audio trace down here to our extracted broadband in real time. And then we can give them different cues, and they might, in a different task, this, again, we're not interleaving nouns and verb generation, but we do have them do verb generation tasks because we want to look at speech representation. And we do this primarily clinically, just as an off-label way for me to identify an off-label in the sense that it's not an off-label intervention, but I'm using customized software to get an idea for functional representation for patients that participate with us. And we use this in conjunction with things like electrical stimulation mapping. Something you guys might find interesting here is we're seeing activity here on the right side of the brain. And this is radiologic right, so it's on the left side of the screen. And this because this patient in particular happened to be have right-sided dominant speech. And we can identify these same nodes in speech networks, very similar to how we did in motor networks with primary and premotor and so forth. And so we can extend these tools based on this intuition for signal that we have. And these tools work in cases of lesional epilepsy and non-lesional epilepsy. This is a case of these are cases of non-lesional epilepsy. You can see we correctly mapped speech and motor to areas where there was seizure onset. Okay, and we can also map function in the vicinity of pathology near things like vocal cortical dysplasias, as you see here, or in the case of the injured brain. So this is a patient who had a stroke. We did stereo EEG to localize where his seizures were starting from after his stroke. And in this patient's case, they happened to be right in the area. The seizures were starting right from an area of hand function where when we stimulated, it both interrupted his hand movement and also induced a seizure. We can correlate stimulation mapping with these task-based changes. We find they're tightly coupled. This manuscript is actually going to be up this week, but I don't have links yet. But for those that are interested, you can contact us and we'll send you the draft when it goes up on archive in the next few days. And again, stereo EEG is very close to what we would expect from fMRI in terms of functional representation, comparing electrophysiologic changes to bold signal changes. We can ask simple questions like, does the motor homunculus, as we've known it for a century from Penfield, extend down into the depths of the central sulcus? This is that same manuscript. And we find that, yes, it does, which I think is interesting. But in addition to these areas that are somatotopically specific, there are also areas of shared representation. So this allowed us to identify what we call the Rolandic motor association area. But basically, there are areas interdigitated within the classic representation for parts of the body on the precentral gyrus that change no matter what part of the body you're moving or what side of the body you're moving. So they seem to have all of the properties we usually associate with premotor areas or motor association areas, but sit down in the depths of the central sulcus. And so we've called this the Rolandic motor association area. And we find it's down in the sulcus itself. The timing of the activity there, which you see here in red, is very tightly linked to the time of individual movements. So it's resolving individual movements. It's not a general planning area. It's actually involved with somehow the generation of individual movements, at least of the hand, foot, and tongue. And the activity of both this association area as well as the primary motor sites precedes and continues through movement, but we can't really resolve time differences between the two. Take-home points for the stereo EEG part are that we can measure from the brain volumetrically. If we're looking at motor cortex, we both identify an extension of classic representation we've understood from the brain surface, but also can do things like identify areas that we didn't know about before. We can do brain-computer interfaces, as I showed you before. I'll show you a couple of examples of these. So here we have a hand area that we're measuring from. You can see he has a left-sided M1. He cheated there by moving his hand to cause that area of the brain to become active, to send the cursor up. And this is just a one-dimensional task. What we're working on is trying to understand how the brain changes in response to feedback, to look at principles of brain-computer interfacing rather than, let's say, going for very high-dimensional control or very tight decoding of a very focal brain region, where we actually want to see how brain circuitry over the broader sense changes during feedback. And we can ask what areas are recruited during feedback. So here we have movement. You can see there's a primary motor area and a premotor area. You can see during imagery that this premotor area becomes more actively engaged and primary motor area becomes less strongly engaged compared to the premotor area. And then during feedback, we have areas back here in parietal that had no activity during overt movement or imagining that movement, but had robust activation while performing the feedback task. And then lastly, I just want to show you an example. I hope the audio works here for you. Doesn't seem to be. But we can have patients control video games. This is more of a fun demonstration than anything that has to do with uncovering new principles. But of course, the more enriching the tasks we have the patient engage in, the more robust we expect the distributed circuitry of the brain to interact with one another, right? So we expect all the circuits to be more readily engaged when it comes to things like reinforcement, reward, and frankly, probably things like error correction when we have robust feedback. And you can see that at least in this one-dimensional task, patients are able to do well. That brings me to the end of my talk. Like I said, our team is lucky to be funded by a number of people. For those that are interested in any of these papers, please get a screen grab here. Most of the talk today was discussing this paper on the top. For anyone who's interested in electrocorticography or in any of the methods from the paper at all, both that paper that I showed you and also sets of, I think, 15 other types of experiments are all in this library of data and analyses. They're all open source. And I would encourage people to download and use the data and the code. I'm lucky to be part of a big team, both here at Mayo Clinic, as well as with collaborators around the country. This is just showing some of the stereo EEG stuff. And then for those that are interested in how we make decisions in stereotactic epilepsy surgery, what role stereo EEG plays in that, but also how we decide, for example, when to go directly to an open resection versus if we have some ambiguity in the agreement between our imaging and electrophysiology and the symptoms patients have. This reference along the bottom, I think, is quite helpful. Anyhow, it's really a pleasure to be able to speak to everybody out there. I'm sorry I can't see any of you, and I'm sorry I got interrupted by the phone call at the beginning. But I think this is really fun stuff, and I'm glad to be part of a vibrant international community trying to help these patients and also trying to learn about how their brain works when we get to work with these patients.
Thank you so much. That was a fantastic talk. And indeed, we had about 115 people attend. We had about 350 register, and they'll all get the emails and the recording to this. So you might have some questions after a little bit of time. So now's the time when we open up Q&A for the audience. So folks, if you have any questions, please do type them in the Q&A area. And while we're waiting for the questions to come in, it was a great last slide here actually that you're showing, because that was going to be my question to you, is whether you could speak a little bit about the differences when you're doing surgical planning when you are using ECOG and SEEG. We do have a question though, so let's jump to that.
Did you? Okay, sorry.
Go ahead, unless you wanted to say something quickly.
Well, I
but
think
that is definitely where my mind is going, is the differences and the implications for you and your practice and how you do resective, the area that the resection should be.
So, I mean, the differences between electrocorticography and stereo EEG that are most relevant, of course, are stereo EEG allows you to really look at the volumetric, and I apologize for looking away. It allows you to look at the volumetric representation in the brain, whereas electrocorticography gives you a very regular view along the cortical surface. So if you're trying to discover and characterize a seizure network, that will often involve areas that are down, let's say in the insula here,
Right.
which we've known play an important role in many patients' epilepsies, but we haven't necessarily understood the role as well, until, as we do now, until we started doing these stereo EEGs. It's important to note that, we started doing it in America about 10 years ago, maybe
Mm-hmm.
12 years ago, but they've actually been done since the 50s and 60s with Jean Taylorac in France.
Mm-hmm.
And particularly in France and Italy, there's been a lot of important work that's been done using stereo EEG for diagnostic purposes, but also therapeutically. After you've identified the seizure network, you can pass radio frequency energy through, do burns in the brain that are very focal. And I think that pioneering work that they've done really made it so the transition in America was easier.
Mm.
Electrocorticography still has a really important role because sometimes, for example, on this brain here, let's say that you have some abnormality here, like either like a malformation of cortical development, like a focal cortical dysplasia,
Mm-hmm.
or you have a low-grade tumor, but you don't know how far forward your functional representation extends. Sometimes we'll put grids here and we can both map the seizures as well as map the function of the brain together. And we do this with stereo EEG as well. We just don't have a regular map like this. And we can look for a margin between the two. And that's the time, I think, most often when we're still using electrocorticography is when there's known pathology that's very clear on imaging. And
Mm.
again, I'm speaking generally. There
Mm-hmm.
are still times when we use grids for other purposes, but when we're looking for a margin between the pathology, meaning the place where we can see a source of seizures that we're fairly confident is where they're starting, but we don't know the extent. And then
Mm-hmm.
on the other side, when we know, well, we think brain function is here, but we don't know its extent that way. And we can find a margin between the two using electrocorticography. Although I think strategies to try to do that with stereo EEG are evolving, and stereo EEG, there will be aspects of that, of finding that margin that will probably be taken over progressively by stereo EEG. I think electrocorticography will have a role for a long time to come yet, but a diminished role. And then we
Mm.
use electrocorticography in the intraoperative setting quite often.
Mm. For
For
mapping.
example, to find that electrophysiologic margin where we think seizure activity is continuing to in the operating room by
Mm-hmm.
performing recordings in the operating room at the time of surgery. And that's where the neurologists that I work with and I will have an ongoing dialogue during the surgery. They'll be down in the operating room with me, and we come to some of those decisions together.
So when I was listening, thank you. And when I was listening to your talk and you were talking about ECOG, if my understanding was correct, that I was understanding that you can calculate the relationship between two electrodes that are separated by a centimeter from one another by calculating phase coherence. So you could make a hypothesis about whether or not two electrodes are involved in the same phenomenon that you're interested in, and then but you wouldn't understand how deep that goes per se. So you talked about volume just now, that SEEG is good for characterizing volume. And so for SEEG, then if I'm following, you would be able to characterize the volume, but then your hypothesis would have to be fairly specific because you wouldn't necessarily understand the the area going laterally or outward, right, from those electrodes.
Yeah, that's true. And again, phase coherence is relevant when we're trying to look at the way that they may have some shared um, influences on them. If
Mm.
the oscillations are really just reflecting that influence that some other brain area, let's say the thalamus, might have,
Yeah.
it doesn't necessarily mean that those two areas are interacting with one another, right? Because
Uh-huh.
they common inputs. And this is a problem we run into in general with these types of measurements and recordings, is whenever you're looking for an interaction, you never know if there's an intermediary, or you never know if there's another site that's contributing to both, introducing correlation. There are metrics also for like looking at the broadband compared with one another. You can take broadband power and do things like Granger causality on that, or you can even take the phase extractions of that of the broadband power, and then use things like coherence of the phase extractions, which is sort of a shorthand, because you don't you're really sort of in fact that because we know those oscillations are causing entrainment in the broadband, that would really just be serving that as a proxy for coherence, which you can measure directly. But in something that's, let's say, an event locked, something where you had visual evoked potential, that kind of thing, then you would want to use something besides phase coherence. And we have lots of tools for that too, but that's talk for another day.
Yeah, that's a different talk.
And those tools are sort of emerging more than all out in the literature.
Yeah. Okay.
But we're happy to share and collaborate. We have a big group with a lot of questions.
Yeah. So we do have, so I'll hold the first question, since that's a pretty broad topic, I would imagine. We'll jump to the second one. It says, thanks, Dr. Kai. I think it's pretty interesting to see the approach of the broadband as a way to take on the general rhythms and also the more deep ones in the brain. Still, I believe that the artifacts in these systems are still limiting to denoise volumetrical approaches precision. They are great allowing us to know areas affected and all, but what do you think would be the future of such instruments? More electrodes, better surface coupling, AI training for denoise, interpret broadband signals?
Okay, let me try to unpack that. So broadband is a way to take on the general rhythms. Well, I think broadband is so if that means that broadband is a way to take on the fact that you have a lot of things going on in the signal, and that's something generic, I agree. But then we're not talking about rhythms. But you can also look at the influence of those rhythms by seeing how they on the brain, or try to infer something about them by seeing how they influence that broadband. And I think that is useful. The artifacts in the systems are limiting to denoise volumetrical approaches precision. So I'm not sure if that segment is getting at a source localization problem. I don't know that it's just the systemic artifacts. I think it's more a matter of you have lots and lots of sources in the brain, and a limited number of channels that you're measuring from, and you can only do so much triangulation from if you're undersampling your system. And they're great at allowing us to know, what would be a future of such instruments. So I do think that there may be utility in higher density of electrode contacts on each lead. But again, if you lower your density, you're then averaging over smaller patches of tissue, and the noise, because you're measuring something that is not phase coherent necessarily, when you're getting these broadband things, then the robustness with which you're going to get a trace that's statistically robust actually increases with your electrode size up to whatever the scale of the representation you're trying to look at is.
You had that slide also where you were showing how the frequency band kind of bottoms out with the instrument limitation. How it sort of it induces the appearance of oscillations when there aren't any, actually, because we can't actually measure as high as we want to because of amplifier limitations. I was interpreting this question to reference this slide.
And notably, this doesn't mean that you can never look at higher frequencies. If you have some synchronized phenomena that's generating a signal that's relatively large in amplitude, like we see like with things with HFOs, that can be up above this noise floor. It's just that this broadband phenomena has that 1/f shape where power is going down very rapidly as a function of frequency. And so it really bottoms out on what the higher range you can look at is. And even if you improve your amplifiers, I mean, the power is falling off like at the high frequencies in the electrocorticogram, like 1/f to the 4th. And so you're not going to, your tech has to get many, many times better just for marginal benefit. But the good news is if it's more or less the same phenomena, then you mostly capture it in this range that's, somewhere above 50 hertz and somewhere below whatever your noise floor is.
Thank you. In terms of your experience with children, which is a question here, you are a pediatric neurosurgeon. Are you able to talk a little bit about the main differences in your experience between working with peds and working with adults as a neurosurgeon?
Uh, well, so as far as these things go, most of the mapping and things that we do, because you can measure behavior passively here in these types of, let's say in these types of situations, speech
Mm-hmm.
mapping, um, all you have to do, not all, but what you have to do is then just have to segment the periods of time when they were speaking and not speaking. So they don't have to be able, they just have to be able to have a conversation with you at worst, right? And so there are a number of challenges that we have with standard mapping approaches, right? They have difficulties with fMRI,
Mm-hmm.
the way that their tissue response stimulation, because the differences in myelination. If they're
Mm-hmm.
younger, it's different than people that are older. So those are considerations. And then and then just your interactions with them. But almost all of my story, or in the this mapping paper that we have coming out, I think 18 of the 20 patients are pediatric. Um, you know, about 2/3 to 3/4 of my patients are pediatrics.
Down to what age?
They'll, I think 5 is the youngest we've done
5.
story.
Yeah,
Yeah.
that was going to be my question. Yeah, is down to what age do you think SEEG is useful and indicated in pediatrics smaller than 5 years old?
Yeah. And, I think a lot of times it can be easier when when they're significantly younger. Um, you know, the relative maturity is different for different children. And, in particular, on
You're
average,
speaking about anatomical maturity, right?
no, no, no,
Oh, okay.
maturity in terms of behavioral,
Okay. Okay.
their ability to tolerate being an inpatient,
Got it.
it's different, based on, and different children are different. And I think on average, young girls are more mature than young boys at that age by some correction factor of a year or two. I'm not sure exactly what it is, but all of these things you have to take into account when you're interacting with the patient and their family. Family support it's important for every patient.
Yeah.
It's even more important for the
For the
pediatric patient.
kids. Yeah. That makes sense. Yeah. Okay. And then another question. Kai, thank you for the interesting talk and the fantastic, inspiring body of work. Do you use functional language mapping in your clinical practice, for example, to select sites for electrical stimulation mapping or directly to guide or limit resections?
I would say we don't use it directly to guide or limit resections. I think it can be helpful to, um, there have been times when we will talk with the neurologists ahead of time and
Hmm.
say we think these are the best early candidate areas, although oftentimes you also know that from where. And so it may have some influence on the order that we do some of the electrical stimulation mapping in. And and we do use it as an adjunct. It's never anything other than, let's say, an adjunct, but it doesn't, it doesn't generally guide, or in fact, I don't think ever it's guided, for example, where we would resect and not resect. But I think for things like what sequence do you start the stimulation in? Because if the patient has a seizure, that can really, during the mapping process, which happens, with semi-regularity, you want to make sure you have the highest yield regions first. And so those
Hmm.
are regions where the seizures are starting from, and the regions, especially if they're suspected to be eloquent, and the regions that are of high eloquence where you have early secondary spread, that kind of thing.
Hmm.
Or
Hmm.
proximity, even if the seizures aren't starting there, they're close to the areas where the seizures are starting from. And we can learn something about the function ahead of time and try to have a little bit better prior on where to start our stimulation mapping first. But it certainly has not replaced anything else or been used instead of anything else. It's an adjunct that we use.
That makes sense. Yeah. Um, Given, you mentioned that stereo EEG has only been around in the in the United States for about 10 years, and in contrast, it's been around in France and Italy for far longer. From
Mhm.
your perspective, can you talk a little bit about what you think the current challenges are in terms of SEEG being used in the United States, and to contrast that, maybe um, some of your hope for the future and where you think the field is headed next, and where you think the field should go next?
Well, I mean, I think that stereo EEG is, because we were able to leverage the experience of the Europeans, is actually fairly mature in the US. But the overall practice itself is maturing very quickly, right? So
Mm.
there are epilepsies that we couldn't treat previously, with resection because they happen to be multifocal or they happen to be eloquent. Um,
Mm.
where you can have combinations, you can do stereo EEG, discover that that happens to be the case, that they're multifocal or eloquent, and try to take out some of the regions of seizure onset or spread with radiofrequency ablation, and that may guide where you do laser later. You can also have, you can do test stimulation to see, well, if they have a lot of seizures, if you're down on their medications and they're seizing a lot and you start stimulating, and again, they have to have a fairly high number of seizures to see a decrease over the time scales in which we do test stimulation, which isn't going to be a prolonged thing, right? We don't want to draw this process out for them. But, you know, having it inform more tailored resections, that's how we've always thought about it. But having it inform stimulation and having it inform laser ablation, and then using as a tool for radiofrequency ablation, those things I think are evolving. And those are actually, it's actually bidirectional, I think, with Europe. I happened to, you know, I my fellowship training, some of it was in Europe at in the Netherlands in Utrecht. And I think that, you know, that there are certain things that people there that use stereo EEG have been thinking about for longer than we have, and we want to get everything we can from that. And likewise, we want to share everything we learn.
Nice. And would you say a little bit about where you hope to see the field go? What's next in terms?
I mean, it's a little bit, I guess, a little bit cliche to say, but, you know, higher channel counts on the same leads. I
Yeah.
very much like the fact that the 0.8 millimeter diameter on stereo EEG is significantly smaller than what we had a decade ago. And I think rates of hemorrhage and things like that have come down considerably because of that.
Right. Yeah.
I don't know. I mean, I don't know that we could go smaller without losing rigidity. Anytime you lose some rigidity in the way that you place these, of course, then you're inaccurate, and there are dangers associated with that. And also you're not measuring from the area that you want to measure from.
Yeah.
So, you know, higher electrodes, which runs counter to having smaller diameters. And I don't know that where the sweet spot is, but really I know that complication rates are exceptionally low with the 0.8 millimeter diameter stereo EEG electrodes that we have. It's not company specific. That's sort of become what the different companies have. And, you know, this direct understanding, you know, we use some algorithms that we have developed for single pulse electrical stimulation. And this is really tied more to research protocols that are on IRBs and that kind of thing, to look at connectivity in brain networks and understanding when two areas that we might stimulate together are easily connected to one another versus when they're not, for customized stereo EEG system implants. I think that's a direction we're moving in that I'm particularly excited about, and that, you know, at Mayo, we get to be part of that. And we have a great multidisciplinary team. We have there's Greg Worrell, who has been doing many things over many years involving novel diagnostic techniques, but also development of new devices, and changing our understanding of all kinds of electrophysiologic phenomena that we measure from these, like HFOs and whatnot. I'm lucky to be able to be a part of sort of the empire that he's built here. We have a great cluster of people. And so we have people that are spanning the engineering disciplines. You know, there's Nuri Inche, Dora Hermes, Brian Lundstrom, Nick Gregg, Keith Starnes. These are all the stimulation team that we have, who are scientists and clinicians. Sorry, those aren't all of them. They are all part of it, but then I'm sure there's people, and if this is on video, I'm really sorry if I didn't mention your name and you should have been mentioned. And I get to be, you know, a role player on a big team, which is fun. I get to, Jamie Van Gompel, who's one of the other surgeons, and Rishna Ali and Rick Marsh and I all sort of support that effort.
Nice. Thank you very
Thank you.
much.
You're
Yeah, that's
welcome.
really interesting, especially to hear about the size of the contacts and how the balancing of that with rigidity and
Yeah, and we're trying to learn about that, and there may be leads that are matched to future DBS constructs.
yeah, right. Right. That makes sense. So lots of good things coming yet. And
Yeah.
so yeah.
And you know, we have a lot of work we've done in understanding evoked potentials and network connectivity, that one of my colleagues or I could tell you about in the future, could tell the audience about, that might be integrated into software.
Yeah, we'd love to hear about that.
All open source stuff. Yeah.
That would be very cool. All right. Wonderful. Well, that's all the questions that we had from the audience today. And I won't take any more of your time since I know you're quite busy, but I do want to thank you very much for coming. This was a very interesting talk, and I hope that we will hear more from you. So thank you very much.
Thank you so much, Marie.
Yeah,
Take care.
you
It's an honor.
too.
Okay.
Thank you. Bye-bye.
Thanks.
Bye, everybody.